Welcome to the members of the Times 3 SOs. In this video, we're going to see about the procedure on how to start our research for the prevention and eradication of coronavirus. Before we start, we have a good news. Japan has invented a medicine called AV-GAN, which is antivirus GAN, that wasn't invented for corona, but for normal flu viruses. After changing its potency during their research, it has successfully started to get corona under control. As we already saw, Cuba has a medicine called interferon alpha 2B. Let's put these aside. Now we're going to use the knowledge treasure that is already available in our country. Using that, we will be able to invent an outstanding medicine for corona. Our ancestors, that is, Siddhas, who are none other than the world's first scientists, for the sake of human race, have given out ideologies about medicines for the diseases that are already there and the diseases that might appear in the future. We can try out those ideologies to invent a cure. So let us keep continuing our research. Independent self-knowledge is the best asset one could acquire. Only this knowledge is found to be permanent and it is found to be expendable. So let's keep that in our minds for our research. Our association consists of scientists, doctors from different fields of medicines, researchers, people with abundant knowledge on anatomy, physiology, pathology, pharmacokinetics, pharmacogenetics, and of course, traditional medicine doctors like Siddha doctors. It has been decided to explain our research in a simple manner because of the diversity of our members. Modern day medical research is conducted in the following way. First, discovering the molecular content of a drug. Now, at this point, nothing is proven. However, about 30,000 researchers work on it in the hope that it might get proven. Then the drug is tested on animals like rats or rabbits. Only about 10 to 15% of the researchers are approved for this stage. Next, testing the drug on human. Only 5% of the researchers are approved for this stage, and the success percent at this stage is 1%. If the testing on human phase gets passed, then the pharmaceutical companies gets approval from the Department of Drug Control to get the drug into the market in an international level. Then starts the production of the drug. To get the drug to the doctors, sales representatives are appointed, and the doctors gets informed about the drug's positive effects, its importance, and the pharmacogenetics of it. And the last step would be that the doctors prescribe this medicine to their patients and it could be availed at the pharmacies. The above mentioned procedure to develop a medicine takes years of research and passing through different stages. Obviously, this procedure would not be helpful to develop a medicine for corona. We are in a state of emergency and we do not have such time to spare. The above-mentioned procedure is followed only because the so-called modern-day medicines are prepared solely out of chemicals, which is a foreign entity on our body. Hence, extensive research on these chemicals has to be done. Its toxic nature has to be studied and tested first on animals, as even a small amount of excess dosage on human could turn out to be fatal. Thereby, these medicines are first tested on animal until they get a positive effect. Along with it, the side effects and the amount of drug to be administered are also identified. Later on, they are tested on human subjects and the amount of drug to be given is adjusted again. Only after these clinical trials are done and the drug in hand succeeds, then the medicine is released. We don't have the need to follow the above mentioned procedures as we are going to use the formula already tested by our Indian siddhas and yogis who are the greatest scientists in the world. We're going to continue our research using this as our foundation. In the next video, we will see about how to successfully complete our research. Jai Hind.